Welcome everyone. Hi, I'm Denise Bonilla. Thank you for being here today. So we have a working group that's working on updating our strategy, our veterinary services strategy for Rift Valley fever. And as we were doing that, we started doing that and started, coming together to be able to pull the correct people in the room. We thought that it would be really nice to spend some time reviewing what we knew about Rift Valley fever as a potential zoonotic vector-borne disease here in the United States, but not just in the United States, although we're worried about the United States. We want to know worldwide what's going on. And so we brought in two experts. We hope two experts, but we do have Terrell here.
On the ballot school board, but only 4 of the candidates are Republican. The Republicans.
Hi, whoever's talking about Republicans.
Hold a second, I'm gonna have to. Hey, Barb, pick it up and hang up.
That you vote for the Republican candidate.
Oh, goodness.
By the Frederick County
Just 1
Republican
sec, sorry about
Committee,
that.
Kelly
It's okay. Okay. This always happens. Don't worry. We're okay. So we're really happy to have Dr. Mike Terrell today here. Once he comes back in the room.
Sorry about that. It was a call. Well, obviously it's a political call.
Yeah.
I purposely didn't answer it, so the voicemail came on.
Okay. Yeah, that makes total sense. So, but I'm gonna for now. I'm going to yes. Thank you, Lee. No Hatch Act violations. Didn't happen. We didn't hear it. So we're gonna move on. I'm gonna introduce Dr. Terrell. Dr. Michael Terrell is a recently retired principal investigator at USAMRIID, which is the United States Army Medical Research Institute for Infectious Diseases. He had served there for more than 30 years. He received his BS and MS in entomology from Cornell University in New York and his MPH from Tulane University in Louisiana. And then his PhD in epidemiology from the University of California at Berkeley. His principal interest has been the study of factors affecting the ability of mosquitoes to transmit various arboviruses. He's evaluated the potential of selected arthropods to transmit pathogens such as Rift Valley fever, Eastern equine encephalitis, Venezuelan equine encephalitis, Japanese encephalitis, and West Nile viruses. These involve both studies in places like Peru, Ecuador, Honduras, Costa Rica, Senegal, Liberia, Kenya, Republic of Korea, Uzbekistan, etc., and then laboratory studies. In addition, he's conducted research on the development of vaccines for several mosquito-borne diseases and the development and evaluation of antigen and RNA detection assays. Like I told you, he's pretty special. He's got more than 200 referred publications in science, the American Journal of Tropical Medicine and Hygiene, Journal of Medical Entomology, Virology, Infection and Immunity, Journal of the American Mosquito Control Association, and others. He's given more than 200 more than 120 presentations at national and international meetings, including invited presentations at more than 20 international meetings. In addition, Mike serves as the editor for PLOS Neglected Tropical Diseases, as a reviewer for numerous journals like American Journal of Trop Med and Hygiene, Journal of Medical Entomology, Emerging Infectious Diseases, Journal of Wildlife Diseases, Journal of Vector Ecology, Malarial Journal, and also the American Mosquito Control Association. He's been recently rewarded the Meritorious Service Award by the American Mosquito Control Association and the Award of Distinction by the American Committee of Medical Entomology. So congrats for an amazing lifetime of service so far, Mike. We're really happy to have you here. And with that, I'm gonna hide myself and let you start on your presentation. Thanks again.
Denise, thank you very much. I'm going to try and keep this somewhat informal. Rift Valley fever virus, what is it, and what is the risk of that virus coming to the Americas, to Europe, to Asia, and to other areas that may cause a real problem? Well, first of all, what is Rift? It's primarily a disease of livestock, cattle, goats, and sheep. In these animals, older animals get sick and they usually recover from moderate viremia. Young animals lambs, calves. More than 90% will die. It is a very lethal disease for young animals. They'll have very high viremia. Pregnant animals, nearly all of the pregnant animals will abort. If they don't abort, almost all of them will die. So there's virtually no next generation for animals. And unlike many zoonotic diseases, this virus, zoonotic pathogens, this virus also affects humans. Most people get infected, have a mild febrile illness. You don't feel well, you're fever, but you recover. A small percent will develop encephalitis. Most people will recover from the encephalitis, and a small percent will develop neurological disease, so will develop hemorrhagic disease, and most of those will die. It is a bad virus. Outbreaks was first described in the Rift Valley of Kenya in 1931. There have been various outbreaks about every 10 to 20 years in sub-Saharan Africa, the southern half of Africa, Kenya, South Africa, Rhodesia, Zimbabwe, with multiple outbreaks over 45, 50 years. In 1977, there was an outbreak in Egypt. It was the first outbreak ever outside of sub-Saharan Africa. They estimated that there were over 200,000 human infections. They reported 598 deaths, sorry about that, from Rift Valley fever, but the estimates were actually closer to 10,000 deaths. It was a devastating disease in Egypt, but still only an African disease. In 2000, for the first time ever, there was an outbreak outside of Africa in Saudi Arabia and Yemen. So it has made it over to the Arabian Peninsula. Well, the basic transmission cycle, the virus will start out as infected eggs, Aedes eggs of some species. The eggs are infected with the virus. The area floods, the eggs hatch. They produce infected adults. The infected adults will then bite cattle, goats, or sheep. They'll develop viremia. Other mosquitoes will bite them, infect other cattle, goats, and sheep, and you get an enzootic cycle going on. If you have a moderate amount of rain, you get some mosquitoes, you have an interepisodic outbreak. No one even notices these because abortions in cattle, goats, and sheep in Africa are common. So if you have a couple of extra abortions, nobody would notice. These just pass completely unnoticed. However, if you have major flooding, then you get lots and lots of mosquitoes produced. You have lots of mosquitoes here, and you have an outbreak. And it's devastating because virtually all of the animals will get infected, which means that virtually every pregnant animal will abort. Most of the calves that were born prior to the outbreak will die. It'll be devastating. Some of those mosquitoes that bite the infected animal can also bite a person and infect the person. And this is a horrible disease for veterinarians because they're called in to take care of the sick animal and butchers as well. And both people who work with animals are at high risk of getting infected directly from the animal, from the blood from that animal. That basically goes back at the end of this, mosquitoes that have fed on the infected animals that become infected will lay eggs. Some of those eggs will be infected with virus. They'll be laid in an area that will be flooded in the future. And those eggs can last for several years before hatching. So if they don't get flooding for 3 or 5 years, those eggs will just sit there. And 3 to 5 years later, when it floods, the virus is reintroduced. And that's your basic transmission cycle. These are affected by virus factors. Does this particular virus replicate well in a vertebrate host? Does it replicate well in a mosquito? Mosquito factors. Is this particular mosquito a competent vector? What animals does it feed on? If it's a mosquito that only feeds on birds or preferentially feeds on birds, it probably is not going to be important because this is a mammal virus. On the mammal, does it produce a high viremia? Does it get sick? These are all factors that affect the transmission. And finally, the environment. The environment affects all of these. Well, why are the Americas, and I say Americas, I could change that to Asia, I could say Europe, at risk? Why are parts of the world outside of Africa at risk? Well, are there vertebrate amplifying hosts? In order to have an outbreak, you need to have the vertebrate hosts that would supply virus for the mosquitoes. Well, cattle, goats, and sheep are fairly common in the Americas and Europe and Asia, and white-tailed deer produce a viremia in the US. So we have lots of good amplifying hosts here. Well, this is an African virus. Can an African virus, is it environmentally permissive in the Americas for this African virus to cause disease here? Well, think of West Nile virus, another African virus, and that virus has done extremely well here. So yes, we are permissive for that. Well, the big question is, are there competent arthropod vectors? Are there mosquito species that are able to become infected and transmit Rift Valley fever virus? Well, I've tested a bunch. Some people in Colorado have tested a few, and there are more than 20 different North American mosquito species that have been shown to be competent vectors of Rift Valley fever virus. So we have plenty of competent vectors here. So if the virus gets here, we have a problem. Well, there is no such thing as mosquito control. There's more than 3,000 different kinds of mosquitoes. Some mosquitoes transmit the virus, some mosquitoes don't. If you control the mosquito control is species directed. If you control the wrong species, you might actually enhance conditions for the actual vectors. We need to know which species to control and when to control them. So when a mosquito ingests virus, so this mosquito has taken a blood meal, the little black dots represent virus. If you test this mosquito, she is full of virus. Any test you use, she's positive, but she's not infected because the virus has not yet infected the mosquito. If she does not have receptors in her midgut, she will digest the blood meal, the virus will be inactivated, destroyed, excreted, and you'll have an uninfected mosquito. This mosquito is said to have a midgut infection barrier, an MIB, midgut infection barrier. But if she has receptors in her gut, when the blood meal is digested and excreted, cells in the midgut will become infected. Some mosquito species, and this is true for virtually all of the mosquito-borne viruses, readily become infected, but the virus never, ever gets out of the gut. It is a gut infection only. It's an infected mosquito, but she cannot transmit that virus by bite. These mosquitoes are said to have a midgut escape barrier, MEB. So they are infected, but they can't transmit. But if the virus gets out of the gut, it replicates all over the place. You'll get 1 million virus particles infectious particles in that mosquito. The virus just goes everywhere, except sometimes there's a barrier around the salivary glands. And sometimes everything's infected except for the salivary glands. And by the way, for mosquitoes, the salivary glands are in the thorax, not in the head. I've read too many papers where people have tested heads so they can test a salivary gland. It's not down there. So this mosquito, although it's full of virus, would have a salivary gland barrier and be unable to transmit the virus by bite. However, once the virus gets into the salivary gland and gets secreted in the saliva, if that mosquito probes for even a couple of seconds, it will inject about 1,000 infectious particles, and the virus is transmitted. So if this mosquito is now a competent vector ready to transmit. So I've done some studies where I've taken hamsters, infected them with virus, fed mosquitoes on them, then tested the mosquitoes to find out whether they were infected or not. And just some results, because you've got to show results in a presentation. Culex tarsalis, which is basically a Western mosquito, Western US mosquito, 79% of them was my estimated transmission rate. They're unbelievably efficient. They are by far the best or worst vector, depending on how you want to. From our perspective, they're the worst vector. From the virus's perspective, they are the best vector I have ever tested. Closely related mosquito, Culex nigropalpus, both of which are vectors of West Nile. Essentially, none of them transmitted. They also have a salivary gland barrier. So it has a midgut barrier, a midgut escape barrier, and a salivary gland barrier. These are worthless for Rift. Anopheles crucians. Well, half of them over half of them became infected. There was a midgut escape barrier. I've tested over 200 anopheles with the disseminated infection virus in their whole body. Not one of them transmits virus. It has an absolute salivary gland barrier, so anopheles are not important. Serophora, very high infection rate, moderate dissemination rate. The Columbiae have a big salivary gland barrier. The Ferox have a moderate salivary gland barrier. So Serophora ferox are a decent vector. Mansonia, they can transmit. The interesting one here is Aedes vexans. I've tested field Aedes vexans from Florida, and 25% have been transmitted. They're a decent, almost all of them became infected. High infection rate, moderately high dissemination rate, moderate cellular barrier, but you get a lot of transmission. You take the exact same species from California, Colorado, or southwestern Canada, tested a large number of them. Transmission rate was less than 1%. Same species. So you don't not only have to know what species you're talking about, you have to know what strain of that species, because different geographic populations can be completely different. Just a quick thing here, just some of the species that have been tested. You can ignore dorsalis. All the anopheles are worthless. Culex nigropalpus, quincke fasciatus, and the vexans from California, Colorado, and Canada, they're worthless. They don't transmit. These, depending on numbers, might be important. Moderately efficient. Aedes taeniorhynchus is a large mammal feeder, very high populations. Aedes vexans from Florida, large mammal feeder. Serophora ferox, large mammal feeder, and these could be very common mosquitoes. Those are potentially extremely dangerous. And then the most efficient in the laboratory, Aedes canadensis, japonicus, Culex dorsale, and Culex inornata. So we have some scary mosquitoes in the US that would be ready to transmit this virus should it get here. Well, how could Rift get to the Americas? Over the last 25 years, there have been 3 major viruses that have been imported into the Americas, basically 3 African viruses, actually, but they don't all come from Africa directly. West Nile virus, which is a zoonotic virus, and chikungunya and Zika, which are both anthroponic viruses. These are human viruses. For chikungunya and Zika, this is a mosquito, human, mosquito, human, mosquito, human. Therefore, somebody got bit by a mosquito, got on a plane, flew to somewhere in the Americas, got off the plane, got bit by a mosquito. It was the right species. It became infected. It bit another person, and the rest is history. Both chikungunya and Zika have caused millions of human infections in the Americas. Well, West Nile virus. If you infect a person of West Nile, person was in Africa, Middle East, got bit by a mosquito of West Nile, they got infected. The viremia in a human is so low, it will not infect a mosquito. That person could come home, get sick, maybe even die, but extremely, extremely unlikely to infect a mosquito. So they would not import the virus. They would just be sick. So what happened here is, and I've seen this in many of my travels, you get on an airplane and the mosquitoes, particularly when you have to go out on the runway, walk up the stairs to get in the plane, this cloud of mosquitoes follows you on the plane. If one of those mosquitoes was infected with virus, West Nile virus in this case, when it got off the plane, the mosquitoes left the plane too. They bit a bird, and the rest is, as we say, history. So zoonotic viruses can get here, but it's much, much more difficult. Well, why are humans a particular risk for Rift? Unlike other zoonotic diseases like West Nile, Eastern equine, Western equine, St. Louis encephalitis, Japanese encephalitis, and a whole bunch of others, where humans may get sick, they may die, they produce extremely low viremias, essentially no viremia, so they don't infect mosquitoes. For Rift Valley fever, humans develop an extremely high viremia, often more than 1 billion, more than 10 to the 9th infectious virus particles, extremely high viremias. The viremias could last for several days. people go back and forth between enzootic, it says endemic, it's really enzootic and non-enzootic regions. I have sat in a Land Rover like this, but it didn't have a roof on top. And yeah, between the elephants, it is unbelievably cool to be in the Masai Mara and watch the animals move around you. Of course, the mosquitoes are biting you too, but who cares? You're getting to see giraffe, zebra, elephants, lions, all kinds of fascinating creatures. It's well worth it. Well, if a person were to get bit their last day in the Masai Mara, the next morning they get up, they get on a plane, they fly back to Nairobi, they change airports, get on a plane, fly to Amsterdam, wait a couple hours, get on a plane, fly back to New York, wait a couple hours, go through customs, get on another plane, fly to Oklahoma. When they get there, they feel like, pardon the expression, they feel like shit. They're exhausted, they don't feel good, they have a headache, they have a mild fever. They have jet lag, and they've been traveling for 40 straight hours, they feel miserable. So it's springtime, they go out in their hammock and take a nap outside, feeding all the local mosquitoes, and they could care less because they're just too sick to worry about it. Virus is here. That to me is the really scary scenario, is a person getting bit by a mosquito right there, and then 2 days later being back in the US or in Europe or in China. Any one of those could introduce the virus. Question is vertical and sexual transmission in humans. There's no data that I'm aware of. I would be surprised if the virus could not be sexually transmitted. Vertical transmission, I would almost doubt it. I would guess that if a pregnant woman was infected, she would abort or she would die. So vertical transmission is, yeah, well, yes and no. It's vertical, and then the fetus would die. So basically, in summary, oh, good, I'm 18 minutes. Rift is a real threat to the health and economy of the world. Humans may play a key role in the transfer of that virus and allowing it to expand its range out of Africa, out of the Middle East to other parts of the world. The vectors, even within a species of mosquitoes, the vector competence can be changed dramatically depending on where that particular mosquito is found. Some mosquitoes are absolutely incompetent and cannot transmit rift. Others are extremely efficient. We need to know which ones are important vectors, and many potential vectors have not yet been investigated. We just don't know. And with that, I'm done. 20 minutes. Questions?
All right, we've got a few minutes to take some questions. We'll just take a few though, and then I think Assaf is on now, so after that we'll get going, and then we'll have some more questions on after when we are finished. Anyone have a question right now? I don't see any in the chat. Okay, let's give them some more time to think, Mike. Yeah, we'll do a question and answer at the end. Assaf, could you unmute yourself and let us know you're around?
Hi, Doris Solander here, if you're ready for a question.
Oh, sure. Well, go
Okay.
ahead.
A couple other places that are of concern to me are the baggage handling areas. Routinely, we had our public health veterinarian a few years ago crawling around to get a bat that came off an airplane in that area. The other is Los Angeles has the two biggest ports practically. And within Los Angeles, there is a neighborhood 10 blocks by 10 blocks that allows livestock. And we've been in that neighborhood twice for Newcastle disease, chasing chickens around. But there are sheep, there are goats, there are cattle in that neighborhood. So we have some of these deep in urban areas. So it's not just, the risk is higher than just passengers on a plane.
Mike, you're on mute.
Again, first of all, there are various places that you have animals living in close proximity to people, but I don't think bringing in a cow, sheep, or goat from Africa to the Americas, I think is extremely illegal.
It's the mosquitoes coming in through the port.
It's the mosquitoes coming through the port that are the problem. It's the mosquito, well, through the port, you have two separate things. One, you have mosquitoes that are on a ship, either the eggs or the larvae or the adult mosquitoes sitting on the ship. But if you're talking Los Angeles, you're probably not at great risk because you're coming from Asia rather than coming from Africa. If you're talking East Coast of Maryland, New York, Florida, Carolinas, Virginia, you'd be much greater risk of a ship coming in. Planes, mosquitoes airplanes bring live mosquitoes in every single day. It's well documented. They used to spray with pyrethroids on the airplane. The flight attendants used to walk up and down the aisles with the spray cans and kill mosquitoes. And that disappeared about 20 years ago. I think there are too many complaints about the pesticide. But I've been on many flights where they have sprayed. If you don't do that, I've caught many mosquitoes on international flights. And those adult mosquitoes coming in are great, I think a much greater risk for Rift Valley fever than is the mosquitoes coming in on a ship.
Okay, that's interesting.
Yeah, thank you both for that. We're gonna move on. I know there's some information, some questions in the chat that we'll hit a little bit later. For now, I'd like to introduce Assaf as you see him. Assaf, would you go ahead and load your presentation so we can make sure that it's working correctly? And Mike, thank you. We'll get back. I'm sure we've got more questions for you later.
Good.
While Assaf is loading the presentation, I'm going to go ahead and start his introduction. So Dr. Assaf Anyamba, excuse me, I'll kill everybody's name, is a distinguished R&D staff member and group lead, remote sensing geographic data science in the geographic data science section in the Geospatial Science and Human Security Division of the National Security Sciences Directorate at Oak Ridge National Laboratory in Oak Ridge, Tennessee. So prior to joining Oak Ridge, he was a principal scientist and senior scientist with the University Space Research Association and the University of Maryland, Baltimore County at NASA Goddard Space Flight Center, where he led teams to develop applications for vector-borne disease early warning systems and global agricultural monitoring, utilizing time series satellite-derived climate data sets to support various federal government agencies, including the Department of Defense, United States Department of Agriculture, Foreign Agricultural Service, and the Center for Medical Agriculture and Veterinary Entomology and Defense Threat Reduction Agency, along with others. So together with Ken Lithicum, they initiated, launched, and operated the Rift Valley Fever Monitor that has served defense and public health agencies for 20 years. He's been involved in various cross-cutting forums, including the National Academy of Sciences, National Academy of Medicine, Vector-Borne Diseases, Human Health and Climate Change. In 2019, the Brookings Institute and the Rockefeller Foundation 17 Rooms on Initiative on Transforming National and Global Epidemic and Intelligence Systems in 2020. The World Organization for Animal Health, the Global Outbreak and Alert and Response Network, and Rift Valley Fever and other vector-borne disease outbreaks. Food and Ag Organization of the United Nations for transboundary animal and plant health, plant pests and diseases, among others. In addition, his work has been featured in various media outlets, including NPR, BBC, and actually in print and in Netflix, the documentary Connected Clouds, The Hidden Science of Everything. He's got over 75 peer-reviewed publications in various interdisciplinary journals. And he holds an adjunct appointment with the Department of Civil and Environmental Engineering through the University of Tennessee, Knoxville, and collaborates with universities through the GEO Health Community Practice. So he received his PhD in geography from Clark University, Worcester, Massachusetts, and has an MA from Ohio University, Athens, Ohio, and BA in geography and economics from Kenyatta University, Kenya. So I hope that worked okay. Assaf, are you ready? We can see your notes on the side if you'd like to share your full. There we go. Welcome so much. Thank you so much for joining. And if you'd like to take yourself off mute, we're happy to hear you start.
Okay, thank you very much for invitation to talk to you today. So I'm going to talk to you about exploiting climate data for Rift Valley fever monitoring and prediction. As Denise mentioned, I have been working on this topic or engaged in Rift Valley monitoring and prediction for about the last 20 years. And that is all. Thank you for Ken. He redirected my engagement, research engagement, after I finished my PhD. So I think without him, I'll not be doing this. I'll be doing something else, probably not very useful. So I wanted to start off by giving this or showing this this graphic, and this is from Ken's research when he was posted in Kenya as army entomologist. So at that time, he was posted there was in 1982-83. That was a period when there was a large-scale drought going on across Africa, from Senegal into East Africa. So he obviously could not be able to do his mosquito work without there being flooding, especially if you consider that vectors of Rift Valley fever usually emerge under flooding conditions. So what they did is shown in this second picture here. You can see they are artificially flooding a dumbo. So they flooded this dumbo, and then after several days, they started harvesting vectors that you can see on the bottom right. And from this experiment, the emergence of first Aedes vectors within 15 days, and then followed by emergence of Culex species of Rift Valley fever about 5 to 30 days. So this is the basic foundation, what we have used and applied in terms of modeling. This work will not be repeated until, let me see here, until in 2018, under natural flooding conditions in South Africa. So you can imagine since that time, there had never been an experiment looking at natural flooding of Rift Valley fever vectors, either using artificial means like Ken did, or using natural flooding conditions. Here we are looking at this area called Meadows in South Africa, where we have been doing some work. And what we try to do here is basically to see if we can replicate, or if we can observe the same series of events in terms of rainfall and flooding and the emergence of different vectors. And what you can see here in blue is daily rainfall. And then the different colors show you different vectors. The first one being Aedes species, and then Culex species following that. So it basically replicates the work that Ken had done. Now, that shows a link to point measurements of climate, in this case, rainfall. But we know that we have now a means of global surveillance. For the last 40 years, we have been measuring sea surface temperatures. We have also been measuring precipitation, land surface temperatures, and vegetation. So we basically have the three, I would say the three fundamental things in terms of ecology and climate we need to understand in order to examine the relationship between climate variability and the emergence of different vectors of Rift Valley fever and of any other disease, be it chikungunya, be it Zika, or any other vector-borne disease. But the important thing is not only in being able to gather this information, but also to find meaning out of it. And the way we derive meaning out of all these massive climate data sets that are running up to 40 years is to compute something we call in climatology an anomaly. An anomaly is simply a measure of the deviation of a particular day, a particular month, a particular year from with respect to its long-term average. So what you see here is an expression of those anomalies for a region in East Africa between 2004 and 2010. And on the left is what we call the normalized vegetation index, which is an indicator of basically land surface greenness of vegetation. And on the right is a representation of land surface temperatures. So you can see if you're examining specifically the period of the end of 2006 to mid-2007, you can see that anomaly that is shown in green for vegetation, about 60% departure from long-term average. And if you look on the other side of it, the land surface temperature, the anomalies are up to about -6 from normal. So you can see the convergence between cooler than normal vegetation conditions corresponding to land surface temperature, and then you can see that the green up in vegetation. And this is anomalous because it represents departures from what you observe during a given season. So these are the metrics we actually use in terms of determining when particular vectors emerge. Like for example, in this particular period, in 2006, when it was drier than normal, we saw the explosive outbreak of chikungunya in East Africa and in the Indian Ocean Islands. While in 2006, 2007, we saw the emergence and outbreak of Rift Valley fever. These particular anomalies can actually be related to what is going on globally. And what is going on globally in terms of variability is this phenomena that is called El Niño Southern Oscillation. The representation of El Niño Southern Oscillation is shown here on the top right, where the red shades represent what is known as El Niño. That is when the Eastern Pacific Ocean warms up and results in the distribution of temperature and precipitation anomalies around the world. And the shades of blue show the La Niña events when the Eastern Equatorial Pacific cools down remarkably, leading to another redistribution of precipitation and temperature around the world. So during El Niño events, you can see here represented in the bottom left, you can see areas that are shaded in green are areas that will have above normal rainfall. Those are, for example, in East Africa, part of Eastern Equatorial Pacific and the southern tier of the US tends to get wetter than normal during El Niño. And then during La Niña, that pattern is reversed. So you see here, for example, South Africa is wet. The Sahel is wet. Southeast Asia is wetter than normal. And this dancing of El Niño, La Niña has been going on for a long time. Reconstruction showed that this phenomena has been there for over 500 years. So it's part of what is known as the internal variability of the climate system. So we can tie these so-called teleconnections or differentials in precipitation to what is happening in terms of Rift Valley fever. And what you see in this graphic here on the top right is what is known as a teleconnection map. And what you see that South Africa is negatively correlated with El Niño events, and East Africa is positively related to La Niña events. That is now shown here in the graphs in the maps at the bottom, where you have got above normal rainfall in East Africa during El Niño years, and above normal rainfall during La Niña years in Southern Africa. And basically those hemispheric difference in the anomaly patterns is what gives you these clusters of outbreaks where you have got epicenters of Rift Valley fever outbreaks located in both in Eastern Africa and in Southern Africa. And then you can see also over the Sahel where the outbreaks along the Sahelian landscape will be the same as in Southern Africa. So how do we go about then doing modeling? The importance of having this long-time series data is that we can actually use it as a proxy for many things. So for example, when we started doing this work in 1999, we determined that most areas where Rift Valley fever outbreaks occur are areas that receive 300 to 900 millimeters per year. And you can see those defined here on the map on the left are shaded in green. And then we also considered what areas of vegetation greenness correspond to that. And in terms of NDVI, that was areas that have got 0.15 to 0.55 NDVI units. And then finally, we mapped that on the historical outbreaks, and we see that if we combine the land cover map, then we derive what we call potential episodic area. So this is where you have what I will call outbreaks, large-scale outbreaks of Rift Valley fever, and you can see that this green area basically describe those particular areas. Recently, we have added to that information on livestock density and then human population density in order to drive the model. And then the most important thing in terms of driving the model is having these what I call dynamic inputs. So inputs from climate, including rainfall, vegetation, land surface temperature, because these are changing over time, and you want to determine what areas have different anomalies that will probably correspond to emergence of Rift Valley fever vectors. So the original model we built was basically a threshold model which determined particular thresholds in NDVI or vegetation. And then what we did is we were running this model as using a moving window technique. So we input in at the beginning, we input in 3 months of NDVI anomalies. And then as we get new information, we drop the first month in the series and we add in the newest month. So in doing so, we are actually able to map what we determine to be areas of risk over the land surface. And you can see in this way, every month we are going to output a little bit different area of risk, because as rainfall season changes over the land surface, you are going to begin to see different areas showing up as risk. So this is the model we use for the 23 years between 1999 and 2018. So this is just a representation of a composite risk map of the areas we map to be at risk and areas where outbreaks occurred. So you can see the green area, which is the potential episodic area that I had alluded to at the beginning. The areas in red show the areas we map to be at risk during different periods. And then the different colored dots are where outbreaks occurred on the land surface. And you see from the first experiment, real-time experiment we carried out was in 2006 and 2007. And you can see that 70% of areas that were identified to have Rift Valley fever in East Africa were areas that we had mapped at risk. In Sudan in 2007 was about 50% of the cases. In South Africa, 30% in 2018, but 0% in 2009, 85% in 2010, 2011. You may ask me why 0%? 0% there is because the area where the case counts or the cases occurred in Southern Africa were in this area that is outside the potential episodic area. So it basically means even though we did a very stringent definition of the potential episodic area, still there are particular ecosystems that we need to take into consideration that probably we don't have a lot of information about. So using this particular information, we actually informed early surveillance and early response. And this is shown here in the graphics on the right, where the red line indicates when we first gave the early warning in a particular country, and the blue line indicates when the first outbreak was reported. So in Kenya, it was about 4 months, Tanzania was about 5 months, and Sudan was about 5 months. Basically, this indicates that there is ample time when a particular early warning is given for there to be preventive measures or control measures to be undertaken. And we put this together in publication of in the Journal of Tropical Medicine and Hygiene in 2010, and we actually elucidated on a number of things that can be undertaken in terms of controlling or being to do control of either vectors or do response in terms of education or in terms of vaccination of animals. And this graphic on the top left actually grew out of that period of 2006 and 2007. We got together with the World Health Organization to put this kind of framework of readiness in terms of response to Rift Valley fever. So we have between 2000 and 2018, our information was utilized by various organizations, including Department of Defense, WHO for Human Health Response, FAO for Livestock Health, and OIE in terms of regulation of livestock trade. And just to show you an impact in terms of the uptake of the early warning information, this is shown here in these 3 pictures. In 2006, we gave an early warning in about May or June 2015. And because of that, the countries, particularly in Kenya, they undertook vaccination of different livestock. And on this particular farm that is near Nairobi, which we have used for a long time as as a farm where we carry out experiments, they did vaccination of their sheep. And you can see on the left, this is actually Ken's hand, even though vectors of Rift Valley fever emerged from the Dumbo on this particular farm, because they carried out vaccination, early vaccination, they didn't lose any of their sheep population. This is compared to the period 2006-2007, which is shown in C, where this particular farm lost 80% of the sheep population because there was no vaccination. Now we have been doing some work with Seth. This is in two publications in 2022 and recently in 2023, in terms of looking at globally what areas will be potential from a climate perspective, to look at what areas have the same expression in terms of the same climate variables, climate variability. And this global map shows you what areas will be a potential of the same variability, and if they have the same vectors, will be a potential risk of Rift Valley fever if it was established in those areas. So you can see a big territory of the US concentrated in that. That is mainly in the southern tier of the US, and in Texas and parts of Florida, and many other areas of the world. And you can see the intersection of those areas with the distribution of livestock population. The livestock we use here is from FAO. So that is just a background to show you what areas would be potentially at risk for localization of Rift Valley fever. The other thing to consider is the variability in terms of rainfall. And this is just an example showing you of the rainfall variability in the US in the period between March and May 2019, compared to an outbreak period in East Africa, which was not March and May 2018. And you can see basically the magnitudes of the departures in rainfall are similar in both regions. And you may ask me, why 3 months? We use 3 months because that is a period that we have seen from many outbreaks that if there is persistent rainfall for a period of these particular months, you are likely to have an outbreak occurring. So what we have observed from these studies over a long time is the importance of climate variability and climate shifts. So from that, what we have seen is you need to have a positive shift in rainfall, a positive shift in terms of the vegetation greenness, and a negative shift in the seasonal temperature conditions. That gives you a nice convergence of what I call bioclimatic conditions that are going to be able to result in emergence of large vectors of Rift Valley fever. So I'll basically end at that particular point, and I will entertain some questions. I have some summary notes, but I think this will come up in the discussion. And I'll leave you by just running this animation, showing you over these many years how the outbreaks have occurred in terms of looking at the NDVI anomalies. So thank you very much.
Okay, thank you, Assaf. Is it supposed to end at February 21st, 2020, 2002, or is there more?
No, we can continue the conversation. I don't want it to be full up. This is just an animation to show the process.
Okay, really,
Yes.
yes, thank you so much. It's a wonderful presentation. So we've got a couple of questions in the chat, and if anyone else would like to add to the chat or raise your hand, we've got 5 minutes to answer some questions. I'm gonna go ahead and go to the first chat question, which I think popped up when Mike was talking, and it's from Lee Constant, and it says, do you think that Culex could overwinter with the virus? And do you think there's an 80s species like Vexans in the US that could have eggs that stay viable for years, like Macintoshi in Africa?
Okay, first of all, Assaf, could you stop sharing your screen so we can see each other again?
Okay.
And now I'll try to answer. Okay, Lee, Culex mosquitoes, which are basically more efficient vectors of Rift Valley fever than the Aedes mosquitoes as far as horizontal transmission. The principal way that West Nile virus overwinters in the United States is by overwintering Culex mosquitoes. So yeah, I'm virtually certain if you infected a Culex mosquito in the fall, that mosquito is going to overwinter as an adult female, and she'll come out in the spring and she'll start the virus transmission again. So yes, I think that will definitely work. And the second question was about eggs from Vexans surviving long periods of time. Well, they do. So yeah, but you know this stuff. Both the Rift in the United States could overwinter fairly efficiently in Culex mosquitoes and very efficiently by vertical transmission in various Aedes species, be it Taeniorhynchus, Vexans, Solicitans, take your pick. Next question.
Well, the next one in the chat, and before I forget, I want to say thank you to Seth Gibson for bringing us all together here today. Thank you very much. We appreciate that. But the next question is, can you speak to the effectiveness of the human Rift Valley fever vaccine? And that's from Joanna Davis.
Well, I've received multiple doses of it. I have good antibodies against Rift. I have never, to the best of my knowledge, actually infected myself with Rift. I think all the studies in animals indicate that it is very, very effective. And you have two different types of vaccines. The human vaccine is a killed vaccine, which requires multiple doses and fades over time. The live attenuated vaccine that they will give to animals has more negative effects, but lasts longer. So I think it'd be much more difficult to use the human vaccine in animals because it required multiple doses, and the cost of going out there and vaccinating each goat or each sheep 3 different times gets to be cost prohibitive.
Okay.
And more expensive to produce.
Okay, thank you. Sherry says great presentations to both of you. Thank you. And then Leah Trapp wants to know how susceptible are wildlife species in the US? In other words, how quickly would the disease become endemic and in which wildlife species? So either one of you that'd like to answer that.
Well, it's been.
I think Mike is well positioned to respond to that.
Well, actually, Lee Concept's probably the best one to respond to that. That Rift, cattle, goats, and sheep, while they're domestic animals, they're all over the place. So a disease only of domestic animals would still persist for many, many years, but we could vaccinate them. But the virus has been shown to produce a high level of viremia in deer. And white-tailed deer are a royal pain pest in much of the United States. They eat my garden. I don't like deer, but they do serve. They would serve as an amplified host, a natural enzootic amplified host. So yes, we have the animals here to allow the virus to persist and cause outbreaks.
Okay, thank you. We have time for maybe one more question.
Hi, this is Sherry. Assaf,
go
I
ahead.
wanted to, I don't know if you can see me. I want to say hi. I haven't seen or worked
Hello.
with you in a long time. Thank you so much, both of you, for the work that you're doing that will assist us in preparing and realizing where our risks might be. especially with the looking globally in your models, Assaf, to help us anticipate when our risks might be higher, especially if we can have some understanding of a potential introduction and see where we need to start looking at our potential risks. But thank you both. And Seth, thanks too for bringing them together.
Let me just add one other thing because it's brought up a little bit about birds. West Nile is primarily a bird virus, produces high viremias in certain species of birds. If you take Rift and you inoculate it into a bird, the few studies I've done is the virus is dead, it doesn't know what a bird is, and it does not produce a viremia. So birds, in all likelihood, not be important for Rift. It is a mammal virus, unlike West Nile, which is a bird virus. It does not produce viremia in mammals.
That's a good point, and thank you so much. We're gonna have to wrap it up. Again, thank you guys so much for being with us today and sharing your knowledge. Really appreciate it. Hope to have you back at some point.
One last thing, you have my email address. If anybody, I have no problem with sharing my email address.
Okay, wonderful. Thank you
So,
so much. Yeah. So if anybody has any questions past today, would like to contact either of our speakers, let me know and I'll get you guys in touch. So, all right. Thanks, everyone.
Thank you.
Take care. Good
Bye-bye.
talking, everybody.